University of California. California, Berkeley, working with Dr. Rosemary Gillespie. And I work in a couple different systems. I work in Sulawesi, Indonesia, and I'm currently doing a fire project in California. So I'm going to touch on those a little bit. I'm really just interested in biodiversity formation and understanding biodiversity through time, as well as how biodiversity is responding to global change. So I'm going to talk about that today, and then a lot of these approaches we're using, kind of next generation sequencing approaches, sharing a little bit of that kind of cutting edge work we're doing with these molecular techniques. So let me go ahead and share my screen. Also, I speak notoriously fast, so please, if anyone wants me to slow down, don't hesitate to put it in the chat. Let's see. Okay. Can we see this?
Yes.
Perfect. So yeah, as I said, I was going to today just discuss really understanding spider biodiversity on a changing planet. And as this audience definitely knows, arthropods make up the mass majority of the global biomass. And this is largely dominated by insects, of course. But as insects evolved, so did their major predator, of course, those being the spiders. And so today we have this large diversity of functional strategies and morphologies in spiders. Currently 50,000 species described, although we know that estimated species diversity is probably closer to over 170,000. And through their coevolution with insects, of course, we have this really, really incredible functional diversity that has emerged just in terms of web architecture, lots of specialized web architecture, but also looking at different active hunting strategies, such as the sparacids or the huntsmen and the celticids. Obviously, the most loved spider being the jumping spiders by most people. And so we know today that they're arguably the most important terrestrial predator, consuming around 400 to 800 million tons of prey annually. So of course, they're really essential in nearly every terrestrial ecosystem. But just like other arthropods and other insects, they are greatly understudied. Total papers right now are kind of estimating total terrestrial arthropod species somewhere between 6.1 to 7.8 million species. Of course, with a lot of the major arthropod orders, we're just kind of at the tip of the iceberg for what we've known about those taxa. More recent estimates with spiders are saying, once again, 170,000 plus species. But like other arthropods, as we sample, we're just consistently detecting new species, and we haven't really reached this plateau for the mass majority of families. So this is something I'm particularly interested in. How do we understand this biodiversity that's just really underexplored at a rate that kind of keeps up with anthropogenic pressure? So obviously global change being a massive pressure right now on a lot of global biodiversity. So thinking about how can we rapidly document diversity and monitor communities to allow us to really understand how different biotic communities are responding to global change. So in the early 2000s, DNA barcoding was proposed as a method to kind of work around the kind of taxonomic barriers that do exist. Of course, taxonomy and systematics is incredibly important, but when we really want to document communities quickly and monitor them, we need to think of a method that paired with taxonomy would allow us to kind of understand what's happening in different systems globally. And so DNA barcoding has now really been adopted widely and is being used in multiple studies to look at whole regional pools of species in different parts of the world. And now we're developing kind of these massive reference libraries such as BOLD, where we can pair those DNA barcode sequences with vouchered specimens in museums. And then when you're conducting one of these studies and you sequence all your specimens, you can then take those sequences and compare them to these databases and really quickly get a sense of those compositions without having to do all the kind of laborious taxonomic work first. So Sanger sequencing was really the method we were using for a long time for the past 20 years to barcode specimens, although this is really, really costly and not very time efficient. And so if you're, we all know that collecting arthropods, you get thousands upon thousands of specimens when you go out in the field, and sequencing all of those just isn't really feasible. So in the last 10 years, we've kind of evolved more high throughput sequencing methods that have really allowed us to process, these hundreds to thousands of specimens we may collect in a really efficient manner and sequence them. And so now we have a lot of automated systems that allow us to perform DNA extractions and amplifications really, really quickly. And then we can use next generation sequencing platforms like Illumina to take all of those DNA, amplify them for your region of interest, and attach index barcodes, pull them together, and then sequence them all at a really, really lower rate in terms of cost than we have with Sanger sequencing. And this has also led to the merging of metabarcoding approaches as well as sequencing of environmental DNA. So especially appropriate for collecting these massive samples of arthropods, metabarcoding has emerged as this method where we can extract DNA from a whole community of insects and take all that DNA, amplify for CO1, attach index barcodes, and sequence. And bioinformatically, we can look at how many species, or a rough estimate of how many species are in a community based on a bulk extraction. And for a while, this was actually resulting in the destruction of specimens, which in terms of wanting to do taxonomic work was obviously a big problem. You might discover new species, but you didn't have the specimens. But now we've really discovered that we can use non-destructive extraction. So you can extract all these specimens in a tube, get their DNA, sequence them, and then from there, if you find there's cryptic diversity or there's new species, you can actually go back to the sample and then perform further work, morphological work on those species of interest. Environmental DNA is now another really common method where we can take samples from soil or water or even trees and sequence all the kind of remnant DNA that's left in the environment. And this is really allowing us to get a sense of whole biotic communities without having to actually sample anything. Of course, in that case, you don't actually have the specimens, but in terms of kind of monitoring for invasive species or detecting novel, whatever it may be that you would like to monitor in a system, this kind of provides an approach for that. So these methods these kind of high throughput methods have really expanded our capability to document biodiversity globally and kind of explore how biodiversity is responding in ways that before were really, really challenging. Of course, collecting thousands of specimens and identifying them takes many years, but now with these methods, we can, within the span of a year, understand what's happening in really biodiverse localities. And so with these methods, we really have been starting to explore some of these global biodiversity hotspots that are largely undersampled, despite knowing that maybe around 40 to 50% of all global biodiversity is concentrated in about 2% of these terrestrial regions. These areas are still kind of untouched, and for many of these kind of less charismatic fauna, such as arthropods. And if we look more closely, obviously my interest being the spiders, we see this is a map from BOLD, from that database of barcodes. We see that many areas in terms of the biodiversity hotspots haven't been sampled whatsoever. So we have most of the records in this database from North America and from Europe, but for a lot of these regions in South America and Southeast Asia, we hardly have any records of spiders from those localities. So as I mentioned, I actually work on Sulawesi in Indonesia, and this is the area I'm particularly interested in from the standpoint of biodiversity and biogeography. This has obviously been a region that's fascinated people for a really long time. And that's particularly because there's actually 4 biodiversity hotspots in this region, all neighboring one another. So each of those has a distinct pool of endemic biodiversity that, on the next island right next to that, there's a completely different set of species.
Okay, okay, that's what I just wasn't sure. I didn't want things to crash.
Oh. Sorry, I'm hearing you. Okay, sorry. So this place is just really fascinating in terms of studying how biodiversity is formed through time, and is really valuable when we're thinking about new biodiversity that is left to be discovered. So I work on Sulawesi, which is the largest and the oldest island in the hotspot of Wallacea. It's this really strange shaped K-shaped island that Alfred Russel Wallace was really particularly perplexed by, because it actually represents a transitional zone between Australian fauna and Asian fauna. So he stated that there's no other example on the globe of an island so closely surrounded by other islands, yet preserving such a marked individuality in its form of life. And so this is largely because Sulawesi has remnant taxa from Australia, such as the cuscus, which is a marsupial and is found in Sulawesi, but isn't found on the really close neighboring island of Borneo, or the kind of opposite trend where there's tarsiers in Sulawesi that are closely related to other Asian fauna, but those taxa aren't found on nearby islands that are more similar to Australian fauna. So it represents this really interesting kind of endemic pool of diversity that is distinct in that it has this kind of mixing pot of diversity. not only does it have this cool origin story, but there's actually really interesting patterns of in situ diversification as well. So within Sulawesi, there's multiple areas of endemism. These were really classically defined from the macaques of Sulawesi, where there's the island is split into these 7 different regions where there's a distinct species in each of those areas. And this is really thought to be associated with the paleogeology of this region. So really fascinating that Sulawesi is a composite island. All these fragments merged from the Australian continental plate and the Asian continental plate, collided together around 25 million years ago. And when that happened, area was exposed above sea level in this kind of archipelago formation. So for about 15 million years, these all the like tan colors represent islands. So Sulawesi for multiple years, millions of years, just existed as this small archipelago. And it was really only around 3 million years ago that a lot of tectonic uplift and sea level changes emerged a bunch of land area and created high elevation mountain ranges all across the island. And so with a lot of vertebrate lineages, we see this reflected in both patterns of colonization and more recent diversification. So the tarsiers are a really good example of this, where they're estimated to have colonized Sulawesi around 23 million years ago, right when the island was formed. But then they had long periods without any kind of record of diversification until much more recently, around 3 million years ago, when this island started to kind of, all the archipelago islands started to connect. And you started getting this kind of rapid expansion of land area, in which then we see really rapid patterns of diversification across different regions of Sulawesi. So some people have called this pattern of super endemism, in which you have species that have a combination of really long branches and really short branches. And it makes the Sulawesi diversity even more distinct than the other surrounding islands, which have different patterns of evolution. So this has been really well studied for a lot of megafauna, but for the lots of the like Babirusa with this really interesting taxon Sulawesi that people have really been interested in studying. But of course, a lot of the less charismatic fauna, the arthropods, the reptiles have been largely untouched. And so I'm involved in a project that was interested in conducting a biotic inventory of Sulawesi, sampling these different areas of endemism, and particularly focusing on taxonomic groups that have been kind of previously ignored for the most part. And so this project is a big collaboration with a bunch of different institutions, both in the United States and Australia, as well as multiple Indonesian institutions. So we have, a team of 30 people that are going out into the forest. We're sampling a really wide range of taxonomic groups, from birds to reptiles, amphibians, small mammals, and then arthropods, and my focus being the spiders. So we go out there for 2 to 3 weeks at a time, build these like little mini field camps that we all work at, and then have satellite camps up the mountains. So we try and get as high as we can on the mountains, but tropical fieldwork often provides a lot of challenges for that. But it's a really rewarding experience, very challenging, but really cool. We were actually able to do some canopy sampling last time. Hopefully something I'll do a little bit more of in the future. But I was the sole arachnologist on the trip, and that was really my goal, was to document as much Sulawesi spider biodiversity as possible and start to understand patterns of community formation for the spiders. So what do we know right now about Sulawesi spider biodiversity? Not very much. There's currently 280 species known from the island, and about 70% of those are endemic to Sulawesi. In terms of genetic data, there's hardly any CO1 sequences available, a lot left to be discovered there. And a lot of the taxonomic literature was just, most of the species were described in the late 1800s and the early 1900s. So that also prevents a barrier to really doing work here, because it's really hard to identify species because of this kind of lack of standardized nomenclature for the descriptions at the time. So there's so much left to be discovered on this island, and it's a challenging place because of that lack of species description. But again, this is where DNA barcoding can kind of get us a foot in the door to understanding what's going on. So I collected on these multiple mountain ranges in Sulawesi, used a lot of standardized collecting methods such as beat sampling and leaf litter collection, and then performed DNA barcoding for thousands of specimens I collected. And so I'll be presenting these results today, which is from 3 mountains across Sulawesi, Puerpupu, Dako, and Ilomata, which are each located in distinct areas of endemism. And so we sequenced 2,347 specimens across these 3 mountains, and that generated 686 unique sequences that we can kind of think of as these CO1 barcodes. Now, as I said, there's very little available in terms of Sulawesi CO1 sequences or any even genetic data for most of Indonesia, but we performed blasts to see, how much can we figure out about these spiders solely from the genetic data. And usually for a species level match, you would do something like above 99%, maybe for a genus, 97%. What we find in terms of the percent match is most Sulawesi diversity that we collected is around a 92 to 93% match, which honestly wasn't even allowing us to get to family level. I'd go and identify the specimens myself, compare them to what we were getting back from our blast results, and they were often wrong even at a family level. So this really to me was just showing how unique the spider biodiversity of Sulawesi was. A lot of it is incredibly diverged from even taxa on nearby islands such as Borneo or Sumatra that has more spider biodiversity in these databases, but we're just really getting low matches. So difficult to work with, but also exciting just in terms of how much unique biodiversity we're detecting. So you can use, of course, this CO1 data to look at some phylogenetic patterns and really start getting some idea of, does Sulawesi serve as this kind of transitional zone for the spider species there? So I took 5,500 sequences from BOLD that were from taxa all the way up from China down to New Zealand. So you get this large range of a geographic range and starting to understand if some clades are clustering with Australian groups while other clades are clustering with Asian groups. And we find this to be the case. So this is an example of a clade that's represented in green, that's from Sulawesi, that's nested among taxa that are from Australia. So seeing a clade nested here seems like there might be Australasian origins for some clades, while other groups like these linophiids, the blue and the green are the Sulawesi taxa, and they're nested quite tightly in this linophiid clade that's from Indo-Malay region. So seeing here some evidence that Sulawesi spiders, similar to other vertebrates, may have kind of that mixed origin, that we see this as a kind of a transitional zone where some Australasian taxa exists as well as some Asian taxa. So cool starting to see these patterns. And from here, very preliminary look, but this can guide us on further phylogeographic analyses where we can kind of look more closely at these clades that are emerging as clades of interest or groups of interest. So along with that, we can take these CO1 sequences and construct what we call operational taxonomic units. Obviously not true species, but we can use them as kind of a species unit to do some more diversity analyses and kind of, once again, kind of understand patterns that are occurring. So in total, from those kind of 686 sequences, we detected 531 OTUs or pseudo species kind of thing, and that compared to what we know about Sulawesi, 280 species, you can see that just with these pretty short collection trips, we're really greatly expanding what we know about Sulawesi spider biodiversity. So that's about every 5 specimens we collected, a new OTU was discovered, if you kind of do the math there. So it's really exciting, just amazing amounts of diversity there. We can also kind of look at population genetics a little bit with this data. So each of those OTUs is constructed of really closely related sequences. And so some of these OTUs have multiple sequences within them that can serve as haplotypes. So we can understand a little bit about the diversity between closely related taxa or species across these different regions. So if we look at the mountains and kind of look at what's shared between them, there was only a total of 14 OTUs or these kind of taxonomic units that were shared across all of the mountains. And then there was a 20 to 27 shared between these mountain regions. And so we were curious, okay, are the OTUs that are shared across mountains, are there CO1 haplotypes within those OTUs? Are we seeing patterns of divergence even between those mountains within OTUs? And we find this was the case. So this is the box plot that shows on the y-axis the number of haplotypes within each OTU. And each of the box plots represents OTUs that are found on multiple mountains versus OTUs that were found on a single mountain. And so you can see here that this was a statistically significant result in that the OTUs that were shared across many mountains had higher numbers of haplotypes and were showing some patterns that each mountain contained a distinct haplotype in and of itself. So some evidence of these kind of mountains serving as areas of endemism, which is pretty exciting. Additional with these OTUs, we can look at compositional differences across mountains using some of these classic diversity metrics. So in this case, we can use beta diversity, dissimilarity measurement. If it's closer to 0, the communities are identical, and it's closer to 1, the communities are distinct. And so this is really using that OTU level diversity as a species and seeing how similar communities are based on those OTUs. So we can perform ordination with these. In this case, this is a plot that's showing if the points are closer together, the communities are more similar to one another, and the farther away they are, the more dissimilar they are. And so the colors represent each mountain. And we can see here that the sites on each mountain are clustering together. So you got all the blue together, the green together, and the pink together. But you do see, of course, that some points are much more close to one another. And these are actually sites that are all at a similar elevation. So this could be actually reflecting that there are taxa dispersing between mountains and kind of maintaining that ancestral niche, rather than showing patterns of plasticity and radiation across a mountain. So as you can see here, it's kind of all the mountains that are around 1,000, 1,500 meter sites. All of those are kind of clustering together and seem like they share a much kind of higher density of diversity than sites at different elevations. And we can look at this in another way. We can take beta diversity, the values, and plot it on the y-axis, and then take the elevational difference between sites and put that on the x-axis. So if one site on one mountain is 500 meters and another mountain is 500 meters, that would of course be 0. And what we see here is that there is an increase in terms of how dissimilar the communities become, the more elevationally different they are. And then of course, we also see that the communities that are the most similar are at these much more similar elevations. So again, this idea that potentially taxa are dispersing between these mountains and establishing in localities that are in line with their ancestral niche space. So using the CO1 data, we're able to, just preliminarily ask a lot of really interesting questions, start exploring if Sulawesi has these distinct areas of endemism for spider diversity, look to see kind of if these transitional zones exist, is Sulawesi kind of this mixing pot for spider diversity? And then we can also look at patterns of biodiversity, cross elevation, and kind of understanding what's going on there. So really powerful tool just to begin understanding the diversity that exists. But we can also use this data, and I think this is one of the most powerful reasons that we produce this, to look at patterns of global change in response to new anthropogenic pressure. So this has been a figure that's been definitely floating around recently about how there's this obviously large impact on abundances of insects globally, and how all these different new global threats are affecting arthropod biodiversity and richness across the planet. So not very many studies for spiders, but we know this in general is a threat to global biodiversity. And in terms of understanding what's happening, again, in these really biodiverse localities where we don't have much species descriptions available and we're getting thousands upon thousands of specimens, it's really hard to kind of keep up with how those communities are changing when we can't even, take a sample and understand what's there. So again, this is where these barcoding approaches, both classic barcoding of single specimens as well as meta barcoding approaches, can allow us to track some of these trends and how whole communities are responding to these new kind of pressures global change. So in Indonesia, one of the biggest threats is, of course, deforestation. These are some maps from the Global Forest Watch that are looking at how forest cover has changed through time. The pink is areas that have been deforested in the last 20 years. And the areas in white are areas that really have been deforested previously in the 1900s. So we can see that there's a lot of forest change still happening today. On Sulawesi, that's mostly in the lowlands. So a lot of areas were already deforested, but a lot of the new deforestation is all in the lowlands, while the higher elevation areas kind of maintain this primary forest. So you get this interesting elevational gradient, which also serves as kind of a disturbance gradient, in which the lowlands are really deforested, but the higher elevations have some of this primary forest. So we can look at some of these diversity metrics, this richness being one of them, and see, is there any association with areas that have been heavily deforested or disturbed? So these are just showing rarefied richness on the y-axis, and each of the different bars is on one of the mountains. And what we see is that Ilamata in the middle in blue has the highest richness. And this is quite interesting because this mountain was actually the least thoroughly sampled. It was a really challenging mountain to work on. And so in total, there's actually only a sample size of 474 in this part of the research, and a really small elevation range from 300 to 1,500 meters. Dako in brown actually has the largest sampling size and the largest elevational span, but still has the lowest richness. So interesting pattern here. And when we begin to explore further, we can kind of tie this to these patterns of deforestation. So Ilamata is in blue here, and it's located in this kind of black splotch here, which is actually a natural reserve. It's the reserve of Boganinani, and it's one of the biggest reserves on Sulawesi, as well as one of the only areas that still has intact lowland forest. Torempupu in green is right next to Lorolindu, which is another large protected reserve, and the mountain range extends into Lorolindu as well. So while we didn't collect in Lorolindu, a lot of the forest is still maintained. Dako in brown, however, is not anywhere near protected areas. And if we look at land cover, we can see once again why there's a potential for this richness decrease on DOCO. So the dark green colors represent really pristine primary forest. The lighter elevation or the lighter green colors are looking at forest that has begun to be degraded or is kind of open mixed forestry. And then the brown areas are areas that have been converted to agricultural land. So the top one is showing the sites at Ilamata, and all of them are located in this dark green primary forest. Tuanpupu is the same way, where you have the sites, you have the sites across this really dark green. When DOCO, on the other hand, all the sites there are in areas of forest that have been partially degraded. And then the lowlands actually are in areas that have been completely transferred to agroecosystem, mixed agroforestry. So in that case, the richness makes a little bit of sense. Starting to see some potential consequences of deforestation on endemic biodiversity. We can also look at the beta diversity values and ask a similar question. Are there differences in across these mountains associated with habitat that's pristine versus habitat that's degraded, which that's a pretty intuitive thing that would happen, but the results do reflect that. So this is comparing Ilamata and DOCO, and the low elevation sites on those two mountains are the most dissimilar from one another. And this is again associated with kind of that habitat shift that's happening. On DOCO, the lowest elevation sites are in this kind of mixed agroforest system, while in Ilamata, those sites are in primary forest. So you're having a large shift in the communities that we're finding, dependent on kind of the conversion of the habitat that's occurring. So just these pressures of deforestation and these land use change are really reflecting on the communities that are just responding, and we're seeing completely different patterns of biodiversity in these sites that are really converted into these mixed agroforest systems. Another major pressure on biodiversity globally are, of course, invasive species. So we're seeing global transportation networks now connecting areas that used to be entirely isolated to large land masses. And this, of course, is resulting in the spread of species to places that before were entirely isolated. And of course, then what we've been seeing in many of these systems is that one introduction can have a massive ecological cascade on the entire network. So if it outcompetes a native plant, for instance, that primary producer will then have this cascade across the whole system. And often this means that it additionally opens up space for more invasive taxa to become established in that network. So it almost creates, through this shift, the ability for additional invasives to become more well established. And so when we're thinking about invasive species, often studies are looking at one or a handful of taxa, but we know it's a whole network that's responding, and entire ecosystems are responding to the introduction of one or more taxa. And so it's really important to think about a community level approach when we're studying the impact of invasive taxa. And with meta-barcoding approaches and some of these sequencing methods, we can do that in a way that we haven't been able to in the past. So we can take bulk arthropod samples from invaded habitat versus native forest and look at how those differ. And we can also do this really cool method that we can actually sequence the gut content of spiders and other predators and see how the diet changes. And so really understand some of these biotic interactions, how that predator-prey relationship changes in invaded habitat versus native habitat. So we were interested in kind of doing a project like this, and we did this in Hawaii on the island of Maui, where there's a really severe invasion of kahali ginger. So this picture on the left shows you this boundary that exists along this fence, where on one side there's native forest that's being maintained and the ginger is constantly being removed. Well, on the other side, there's this really dense forest of ginger that has outcompeted the mass majority of native kind of low ground covered plants. So this is a large problem in this area, and we were really interested to see how native arthropod taxa are responding to this invasion, and using this as a little bit of a natural experiment where you have these contrasting native forest and invaded habitat. So we were thinking about this in a little bit of like a source sink context, in which you have a source population of native taxa that may be entering this invaded habitat and having these kind of cascading consequences due to that the structural changes in that habitat. So we wanted to see specifically how endemic taxa are responding. And so we decided to focus on this endemic genera of spiders, Pajupalis. These are in the running crab spider group. And we wanted to see how their diets are changing from ginger, invaded habitat, and native forest. So this gives us a little bit of a potential to look at shifts in biotic interactions and see if there's a really severe impact of this kind of plant invasion on how the spiders are functioning. So again, we did this by, we take the abdomen of the spider, you can extract the DNA, you can amplify your barcode region, and then bioinformatically, you can say, let's just look at the insects in that DNA, and then you get an idea of what those spiders are eating. And we did this by collecting spiders along transects. So the purple lines are in ginger sites and the green lines are in native forests, collected those spiders and performed this kind of gut barcoding. And so what we found here is that there was a large overlap in dietary communities of these spiders. So the plot on the upper right corner shows that, once again, an NMDS plot where the purple spots are all the spiders in invaded habitat and the green are all spiders in native forest. So you can see that there's an overlap there. But when we begin to look more specifically at what is being eaten, we begin to see a large difference here. So the native spiders, or the spiders found in native forest, are in this green on the bottom. And we see that their diets are really dominated by hemipteran taxa. So about 72% of the spiders in native forest were eating hemipterans, followed by lepidopterans and dipterans. The spiders from the invaded habitat, we see kind of this interesting trend of kind of like dietary dispersal, in which there's a kind of a mixed dietary strategy where they're not solely eating hemipterans, they're also eating lepidopterans, dipterans, and particularly interesting is that the spiders in the invaded habitat began eating high numbers of collembolin. So in the native forest, we only found a single spider eating collembolins, while this was really common in the invaded habitat. And this seems to be partially this kind of idea of the invaded habitat allowing space for more non-native taxa. So the collembolins of Hawaii are entirely non-native and are being consumed in high rates in the invaded habitat and not really being found in the diets of the spiders in native forest. Another really cool thing that we can do with this data is we can actually not only detect the prey that the spiders are eating, but also detect parasitotypes of those prey. So this was something we thought we might be able to do, but weren't sure. And we ended up being able to detect both entomopathogenic fungi and parasitoid wasps from this data. And so all these seem to be more specifically related to the prey rather than spiders. But what we found was there was a much higher density of infection of entomopathogenic fungi in the invaded habitat than there was in the native forest. So there was only a single spider that we detected DNA from this fungi in native forest, while there were multiple spiders where we found this kind of entomopathogenic fungi in the invaded habitat. Similarly, with these parasitoid wasps, there were much more detected in spiders in native forest, but we did find that they were significantly higher in the invaded habitat. So again, it seems like that these prey items are being parasitized at higher rates in the invaded habitat. So what this does for the spiders, not really sure, but it does give us some indication that arthropods within invaded habitat are suffering from a higher kind of parasitic load that could be a really negative cost and make this habitat somewhat unsuitable for taxa that exists there. And so the last part I want to touch on is natural disasters and how metabarcoding approaches and these approaches allow us to really rapidly track how communities are responding to natural disasters. So this is a little bit pivoting away from spiders, and this was kind of whole arthropod sampling. But as I mentioned, we're doing this project in California where we're looking at how arthropod communities are responding to wildfires. And so obviously California wildfires are really, really becoming a problem. Fire regimes are changing. And the past two fires, in the August complex in 2020 and the Dixie fires in 2021, were just really massive and burned nearly 1 million acres in both cases. And actually, the most severe large wildfires, the eight most large, were all in the past 5 years. So we're just seeing that fires are getting a lot bigger, they're burning for many more days, and they're getting hotter. So this kind of shift in fire regimes are happening. So we paired with the UC Reserve System to look at sites in both burned and unburned habitat across 6 different UC reserves. This was a total of 76 sites. And as I said, they represented kind of paired sites of burned and unburned. And this allowed us to compare how are communities differing in the burned sites from the unburned sites. We also wanted to look at different habitat types. There's quite a lot of research in forest or grasslands, but there's multiple, of course, habitat types in California that are now being impacted by fire. So we also sampled across different habitat types. And then lastly, there was 2 different time points. So The first sampling occurred in the fall of 2020, and then a second sampling occurred in the spring of 2021. So this allowed somewhat of a recovery trajectory, but also for the arthropods, of course, that's just going to be a large seasonal difference between those two time points. So in total, we had 3,600 specimens that we were able to sequence from this project. And this also just gives a really good example of how quickly we can process specimens. The specimens were collected, as I said, in 2020 and 2021. I received the samples in summer months of 2021, and we were able to get sequences back in November of that year. So those 3,600 specimens, we were really quickly able to see what's happening in this system. And this allows us in the future to continuously monitor these sites and understand how they're responding following fire. So we can first think about species richness. Is there a richness difference between burned and unburned sites? We actually don't detect any kind of pattern of richness differences. This is well supported in the literature, in which there actually doesn't seem to be much of an impact on the species richness. But we were really interested to see if there's compositional differences. So are the burned sites containing an entirely different pool of species than the unburned sites? So again, we can do an NMDS ordination plot where we take all of the sites and look at how similar they are. The red represents burned sites and the gray represents unburned sites. And so what you can see here is they kind of entirely overlap. So we really didn't detect any difference at this point. But this data actually includes everything. So this includes the sites in 2020 and 2021, all the sites across different reserves, et cetera, et cetera. And so what we started to realize is that the largest compositional differences was actually just due to seasonality. So you had sites just being more similar in fall than they were in spring, which makes a lot of sense, especially with pollinator communities emerging in the spring that just aren't in the fall. So we realized we needed to kind of fix this before we could detect any patterns of burn versus unburn. And so this also is a problem with some of these other features as well. So through autocorrelation, of course, communities within the same reserve will likely be more similar to one another. And then additionally, in the same habitat type. So for instance, burned grassland compared to unburned grassland may inherently be more similar than burned grassland to burned scrub. So we wanted to kind of correct for those things. And so instead we looked at the burned and unburned sites if they were in the same reserve and in the same habitat type, and then looked at it across these two seasons as well. And so this is where we started to actually detect some cool patterns. So these are box plots, again, showing beta diversity. The one on the far left is comparing the burned sites to the unburned sites. The one in the green is comparing burned sites to burned sites. And then the one in the blue is comparing unburned sites to unburned sites. So these little pictures trying to kind of demonstrate that. And so what we see here is that the beta diversity between these burned versus unburned sites is the highest absolutely in terms of compositional differences. And here there's actually multiple sites that are entirely dissimilar, so have this value of 1, showing that there's really nothing shared between these sites. If we look a little bit more closely at different habitat types, it gets even more interesting, where we start seeing that the habitat type itself and how that habitat is recovering from fall to spring following fire is going to influence, of course, how the arthropods respond. So for grasslands in 2020, we see kind of that there's the burn sites cluster into this one area while the unburned sites are a little bit more spread. But in the spring, we actually see a different pattern in which the communities in the burn sites actually become more dispersed. So their composition is just kind of radiating and not responding in the same way as the unburned site. So we get a little bit of a different response based on the habitat type we're looking at. We also see this for scrubland, where in 2020, following the fires, there was quite a lot of overlap in terms of the composition between burned and unburned sites. But actually the recovery trajectory into the spring shows something completely different, where the burned sites are a lot more similar to one another and aren't very similar to the sites that are in these unburned areas. So just beginning to kind of look at these results, but we're starting to see that when we look at kind of general patterns of diversity or general patterns of composition, we don't detect very much. But when we start kind of getting in the weeds of it and looking at these differences across habitat types or within reserves, some really interesting patterns are emerging across these different habitats. So just kind of wrapping it up, obviously these methods I think are really powerful in allowing us to begin to document biodiversity in these places that have been historically really understudied, and allow us to respond in a way that we haven't been able to global change. And so allowing us to monitor communities in ways that we haven't been able to in the past. And the FIRE project is a really good example of that, in which we're able to, within the space of a year, collect the samples, sequence the samples, and start understanding how they're responding in a really short amount of time. So yes, thank you for listening and inviting me. Hope I didn't speak much too fast, but I'll stop sharing my screen now. I'd be happy to take any questions. Yeah.